The present status of pharmacognosy, a high-tech frontier. Today, pharmacognosy has completely broken free from its historical reputation as a purely descriptive, dry botanical science. It is no longer just about pressing leaves into herbarium sheets or identifying crude drug adulteration under a basic microscope. In the modern pharmaceutical landscape, pharmacognosy is a highly interdisciplinary, tech-driven field that merges molecular biology, advanced analytical chemistry, bioinformatics, and ethnopharmacology. It serves as the definitive bridge between ancient traditional medicine and futuristic drug discovery. 1. Advanced analytical tools and standardization. The biggest challenge with natural products has always been variability. Plants change their chemical profile based on soil, weather, and harvest time. Modern pharmacognosy solves this with unprecedented analytical precision. Metabolomics and chemical fingerprinting. Using techniques like HPLC, LC megasiemens per megasiemens, and NMR spectroscopy, researchers don't just look for one active ingredient, they map the entire chemical profile of a plant extract. High-throughput screening, HTS, robotic systems screen thousands of natural extracts against specific biological targets, e.g., cancer cell lines or specific enzymes, in a fraction of the time it used to take. Quality control. Regulatory bodies worldwide now demand rigorous standardization of herbal medicines using specific active markers, ensuring that a natural capsule bought today has the exact same efficacy as one bought next year. 2. Reverse pharmacology and traditional knowledge validation. Instead of starting from zero in a lab, modern pharmacognosy heavily utilizes reverse pharmacology. Scientists look at documented, time-tested traditional systems such as Ayurveda and traditional Chinese medicine (TCM) and work backward. Ethnobotany-driven discovery. Researchers identify plants used successfully by indigenous populations for centuries. Scientific validation. The mechanism of action is then studied at the molecular and genetic level to understand why and how it works, transforming experiential wisdom into evidence-based medicine. 3. Expansion of horizons beyond terrestrial plants. While land plants remain crucial, pharmacognosy has expanded into extreme environments to find novel chemical structures that terrestrial organisms simply cannot produce. Marine pharmacognosy. The oceans are a massive focus area today. Organisms like marine sponges, tunicates, and deep-sea microbes live in high-pressure, toxic, or low-oxygen environments. They produce unique secondary metabolites that have led to powerful new drugs, particularly anti-cancer agents, e.g., trabectedin, eribulin, and antivirals. Endophytic microbes. Scientists are heavily researching endophytes, fungi or bacteria that live inside plant tissues without causing disease. Often the endophyte is the actual producer of the medicinal compound, previously attributed to the plant itself. 4. Synthetic biology and plant biotechnology. The modern era recognizes that harvesting tons of rare or slow-growing wild plants is ecologically unsustainable and economically unviable. Elucidating biosynthetic pathways, pharmacognosyists map the exact genetic and enzymatic pathways a plant uses to synthesize a complex molecule, like a specific alkaloid or glycoside. Metabolic engineering. Once the pathway is understood, those genes can be transferred into fast-growing microorganisms like E. coli or baker's yeast to brew the pure plant compound in bioreactors, saving natural ecosystems from overharvesting. Plant tissue culture. Using micropropagation, hairy root cultures, and callus cultures to produce high-value secondary metabolites under controlled laboratory conditions, completely independent of geographical restrictions. 5. Regulatory evolution and global market trends. The global demand for natural products, nutraceuticals, and functional foods has skyrocketed. This has forced regulatory frameworks to evolve rapidly. Integrative medicine. There is a massive global shift toward combining conventional medicine with validated natural therapies. Strict regulatory frameworks. Agencies like the US FDA, with its botanical drug guidance, the EMA, and India's Ministry of Ayush, have established rigorous guidelines for safety, toxicity, and clinical trials of natural products, elevate the credibility of natural drugs on the global stage. Summary of the modern shift. From traditional pharmacognosy to modern pharmacognosy. Macroscopic and microscopic identification, genotyping, DNA barcoding, and metabolomics. Morphological description of crude drugs, isolation, structural elucidation, and molecular docking. Purely terrestrial plant focus, marine microbial and endophytic screening. Wild harvesting and cultivation, tissue culture, bioreactors, and synthetic biology.